Yeah, good afternoon, ladies and gentlemen. This is another opportunity to still meet for another lecture, and today's lecture is actually scheduled to take place for just a few minutes. In the last video we had, we talked about some of the major. Okay, so we talk about this course being a twofold course, which is always on immunology and public health. Immunology and public health. So, just like I said, that major of our discussion has been centered on immunology, and we crown it up, our aspects in immunology, we crown it up with knowing what are the techniques, major techniques in immunology. And after looking at that, where I told us that we have ELISA, which actually means enzyme linked immunosorbent immunosorbent A4 acid. So, and I said that this is a technique that believes in antigen and antibody reaction. I told you that antigen will always come from pathogen in form of any parasite or microorganism, whereas antibody will always come from the immune system. That is, why this one is willing to destroy the body system, this one will be willing to protect you from the activities or disruptive ability of any antigen. And so far so good, I have actually divided this immunology into three aspects. I said the immune response, the first one is the one that is natural, that is innate immune system, and the intermediary, that is what is called the complement system. Then the last one, which is adaptive immune system. So another word for innate immune system is non-specific. Non-specific, that is the one that doesn't have any prior knowledge, any prior experience. And I said about complement system, I said complement system is a cascade. That was part of what we discussed in the last lecture. It's a cascade of proteins. It's a cascade of proteins. And what do all these proteins, what do they do? They are here to complement the efforts of adaptive as well as a specific immune system. And just like I said that adaptive immune system is known for two things. The first one is that through the process that is called clonal expansion, antibody will be produced by B cell, then followed by memory cell. That is, once you have an attack from antigen, if antigen is released, if the adaptive immune system is going to come into play, the first one is that it is going to involve clonal expansion by B cell. Then immediately after that, the B cell will now be recruited in form of antibody to go and fight against the antigen. And once it has encountered an antigen, there is going to be antigen antibody reaction. Apart from that, there is going to be it could lead to elimination of the invading microorganism. Then apart from eliminating it, some of the adaptive immune system is going to store it in their memory, so that whenever they encounter such a an insult again, they will be able to remember what to do. So these are part of what we have done, and specifically in the last lecture as well, we examine what are the chemical perspective of an ABO blood grouping, as well as what is called ABH blood grouping. Not only that, we also look at the the genetical basis of ABO blood grouping, as well as ABH. Also, the last thing was we also look at what could be our immunological explanation of ABO blood grouping. And I remember that I told us at that time that the only difference between ABO blood grouping is that H, that is O, is like at the red blood cell. This is immature red blood cell, and on the surface of red blood cell you have attachment of some carbohydrates, that is in form of sugar. So I think I remember I told you something like that, that if somebody is H group, H is also the same thing as O. So if somebody is H group, this one is going to be made up of glucose, if glucose is here. Apart from that, here the second one is going to comprise of galactose. It's going to be galactose. Then this one is going to be NAG. What is NAG? That is N-acetyl glucosamine. So then the last one, which is galactose. This one is galactose. So on the surface of the galactose, it is attached another sugar which is called fucose. So this is for somebody who is O blood grouping, or we can call the person H blood grouping. So if somebody belongs to O, so this is the biochemical explanation of somebody with O blood grouping. Then now, if we now have somebody who is either A or B, that is still talking about, this is also going to be glucose, it's still going to be galactose. This is going to be galactose. This is also going to be NAG, going to be NAG as well, that is N-acetyl glucosamine. Then on the surface of NAG is attached glucose, is attached galactose, just like this. And you now have fucose. But in addition to this, in addition to H, the next thing will be that on galactose there will be another sugar that will be attached. If this person is blood group A, in particular, if it is blood group A, that means this is going to be galactose and N-acetyl. Galactose N-acetyl. Okay, so that means this person is blood group A. So if this person is blood group B, if this person is blood group B in particular, so that is telling us that this is red blood cell and this person is blood group B. So you are going to have everything you have on H. Everything you have on H, that is attached to it is glucose, attached to glucose is galactose, attached to galactose is N-acetyl glucosamine, then attached to that is galactose and fucose. But and Fucos. So this person is now somebody with blood and Fucos. So this person is now somebody with blood group B in particular. And if somebody is now blood group AB now, because you have some individuals whose blood group are A, blood group ABO. So as I told you that ABO blood grouping was discovered in 1900. It was discovered in 1900 by a scientist called Carl Landsteiner. And immediately after his discovery, there are other type of discoveries that have actually come up, and these type of discoveries have been that they discovered a lot of ABO blood grouping and some others as well. So to tell you that we have some kind of development immediately after that. Then also I told us as well that genetically speaking, that is biochemical. Everything we have done, I have explained here, is the biochemical basis of ABO blood grouping. The genetical basis is such that I told us that the gene that control ABO blood grouping, that gene is located on chromosome what? Chromosome 9. I told you, is located on chromosome 9, and it is on the short arm of that chromosome, which is on locus 34. I told you because I wrote it for you. 9 34. Abi? Yes. Q 34. This is how it is written. It is located on chromosome 9, and Q means it is located on the short arm of that chromosome. And on that short arm of chromosome, it is now located on locus 34. That is the genetical basis of it. And apart from that, ABO blood grouping, that is telling you that ABO blood grouping is hereditary. Somebody who is AB marrying another person who is B, you can determine the type of children that they will give birth to, and that is exactly what is called the use of ABO blood grouping in paternity or in resolving paternity disputes. So I think I remember I also gave you an instance. If a father is AB, and the mother is B, and they are now giving birth to somebody who is O, that is, how can you explain it by your understanding of ABO blood grouping from using biochemical perspective? You should be able to know, and you should be able to even predict the type of transfusion, blood transfusion that they will that can actually transfuse blood for them. That is the genetical basis of it. So then, apart from that, I also told you that the genesis of this means that it's something you can pass from parent to the offspring. Something you can pass from parent to the offspring. Not only that, I also told you that genetically speaking, don't forget that in human there are 23 pairs of chromosomes, and that is telling you that one will come from your father and another one will come from your mother. So that is the genetical basis of it, that it is a kind of blood grouping that has genetical basis. That is, you must have inherited it. Whichever blood group you belong to, your blood group must have either come from your father or must have come from your mother. So that is the genetical basis of it. And I also explained the immunological basis. I told you that whenever you have whenever you have whenever you have anybody who is either A or is either B, so it is antigen antibody reaction. It is antigen antibody reaction. That is telling you that for somebody who is blood group A, blood group A will prefer to take blood from somebody who also has A. So that is immunologically, wherever you see negative, that means, because if you are, it's just like somebody who is taking blood from somebody who is not related. So related plus related will give you no problem, and anyone that is not related, that means there will be agglutination. And I told you that that was the genetical basis of antigen antibody reaction. That's why when you want to do ABO blood grouping test, they will give you antiserum or antisera. You collect the blood, the person's sample, so, and you start adding it. Anywhere you see agglutination, that means the person will not be able to donate blood for that group. So that is the immunological basis of ABO blood grouping system. So, and also the last lecture, I also gave you an example. Another thing, I told you that most of us are only familiar with ABO blood grouping. So if somebody says you are O positive, some of us don't know exactly what it means. If they tell us that we are A positive, we don't know what it means. So, and there is difference between, you can see if they say you belong to blood group O, don't just rely on that information that you belong to group O, and that is to tell you that you can give blood to somebody who is O. No, if you are O positive cannot donate to O negative, there is going to be problem. And what do we mean by either positive or negative? I also told you on Saturday, I think on Saturday, I also told you that that is telling us about a different thing entirely, and that is Rhesus blood. Rhesus blood. I told you that Rhesus has its origin from monkeys, and whatever happens to the monkeys, that means we can actually predict that that is going to be what's going to happen to human. So somebody who is a O positive now means that the person, on the red blood cell of that person, the person is having biochemically something like this. But on another red blood cell or white blood cell, that means that person is Rhesus positive. That is, if somebody who is O positive now marries somebody who is O negative now, if two of them marries during pregnancy, if this is the mother, during pregnancy, the two of them will give birth to somebody that will be alive. The child will still be alive. But what it means is that because the man is positive, within the first 12 hours, they will ask the person to take an injection. And that injection will mean that it is going to save that mother from having a successful subsequent pregnancy. So if the person does not take that kind of injection, second born might not even be alive. So they will have, that is what they call in Yoruba, they call them abiku. So because they do not care to go for rigorous screening, they just think because they are O, they can just marry. There is a difference between somebody who is O negative and somebody who is O positive. O negative means that you are blood group O, which we know, but the rhesus factor, you have the rhesus factor is also positive. So, and that's the reason why you must not marry another person who is also O positive. Although two of you are O positive, you are O, but because you have positive in your, in fact, somebody who is O positive cannot even marry, because if they marry, if he marries somebody who is also O positive, thinking that they are similar, no, what is going to happen is that even the first pregnancy, there's going to be problem. But because the mother is O negative, the first pregnancy, there won't be problem. It is only the subsequent pregnancy, the second pregnancy, the third pregnancy, and the more the pregnancy, the more the danger of giving birth to stillbirths. So we mentioned something like that in the last class we had. So then apart from that, I also defined, I told us that if somebody asks you to differentiate between ABO or ABH blood grouping and the rhesus factors, I told you that the ABO blood grouping is something that has to do with red blood cell, whereas that one is talking about what? White blood cell. Although sometimes it can also affect the red blood cell as well. Then apart from that, you can see now all these are carbohydrates. ABO blood grouping is talking about carbohydrates or sugars. But this one is a kind of protein. In the last class, I think I draw it for you. That is like you have a 7 loop. 7 loop. 1, 2, 3, 4, 5, 6, 7. 7. That is located on the surface of the red blood cell or white blood cell. So it is because of these 7 location of this blood 7, which is protein in nature. It is protein in nature. Don't forget. So somebody who is A positive now, A positive will tell you that on this red blood cell you have the location of all this A one side, and on the other side, which is protein, you have something that is like this, that is either the person is, if the person has D, capital D, that means the person is Rh negative. If the person is having small d, that means the person is D positive or Rh positive. So, and apart from that, this one, ABO, is on chromosome 9. I told you, it's on chromosome 9, which is on short arm of that chromosome. Whereas WBC, which is Rh, is chromosome 1. So you can see that the location of each of these will actually go a long way to determine the type of whether the person is going to be A positive, B positive, or whatever, what have you. So without wasting our time, I think I want to really stop here today so that we'll be able to consider. Even in the last class, I remember I also talked about autoimmune diseases, AID, and I gave you, I think, seven different types, and out of that seven different types, I remember I mentioned them and I cited some case studies on that. I remember I told you autoimmune diseases are diseases that instead of the immune system to fight against the enemy, the immune system is fighting against itself. and that is why they call it auto. Auto is self, automatic. For example, if you have an automatic car, now that means the car that just operates by itself. You don't need to start changing gears or this one. It will just go. So that is why I said autoimmune disease, that is self, the self, and it is one of the characteristics of adaptive immune system. If there is any problem with adaptive immune system, that is telling you that this arm of immune system will not be able to differentiate between self and non-self. And I gave you this like a rheumatoid, the nature has actually made it. And I also gave you cited a typical example case study of an actress that came out and told the whole Nigerians that he has been diagnosed with a special autoimmune diseases that she's going to die very very soon. So it is that I mentioned systemic lupus erythematosus, as well as multiple sclerosis, diabetes mellitus 2 as a typical example, Hashimoto thyroiditis, celiac diseases, as well as other kind of disease. And this is a case whereby I said self inability to differentiate self cell from non-self, and that is autoimmune diseases. So don't forget that that is the immunological aspect of this course. The other aspect that has to do with public health is a very broad topic. You can see that people do spend 4 or 5 years, even in university, to go and learn only public health. So we cannot cover all, but what we will do is just to make sure that we just touch them before we will go for exam or whatever we want to do. But it is very, very good that we have very good understanding, basic knowledge about all this, especially autoimmune diseases. And don't forget that what we have done today is to look at the biochemical basis of ABO blood grouping as well as ABH, the genetical basis, which I said the gene that control ABO blood grouping is located on chromosome 9 on the short arm, and it is found on locus 34. That then I said is also hereditary because you inherit it from both parents, either from your mother or from your dad as well. Then we also look at the immunological basis, which I said it is antigen antibody reaction. And let me just say this to round up. Whenever you have an infection, whenever there is infection, any type of infection, that is telling you that the foreigner or the stranger has overwhelmed, has been able to capture your immune system. And that is the reason why you have an infection. And when you have an infection and you recover from it, that means your immune system is able to fight against the invading microorganism. And that's why I said the relationship between microorganism and human is such that it has to be cooperative enough. It is competitive. And whenever there is competition between the host and the invading microorganism, whichever, it could go either way. If your immune status is so strong, you just discover that you are going to be a state of equilibrium, you have no problem. But once the population of the invading microorganism, in particular microorganisms, they are more populated than your immune system. So it's like you have there's going to be problem on that. So I don't know if there is any question or comments. Okay, okay.
Can our body system fight against
Very, very, very possible. It's very, very possible. Naturally speaking, the nature has endowed us to be able to fight against whatever pathogen that we can actually talk of. And that is the reason why we have the adaptive immune system. The adaptive immune system is such that is quite different from innate. Innate is what we are born with. Innate means that it doesn't have any experience. That means if the invading microorganism is more potent, more powerful to overcome the innate immune system, that means our immune system is going to surrender. But unlike what you have in adaptive immune system must have a prior experience. It must have been able to experience such kind of insult from pathogen, and that's the reason why getting exposed will give us more experience. And that's why I cited an example that children that develop from villages, they are at better way of fighting against infection than people or children that are from the cities, because in the villages you are going to be exposed to a lot of health challenges, and these health challenges is going to make you more wiser. It's just like somebody that is if we are talking of heartbreak, for example now, somebody that has experienced heartbreak 3 4 times, in case another boy is just come and see once I was blind, but now I can see. You cannot deceive me any longer. So the same way, so that heartbreak, somebody that has a lot of experience in heartbreak is just like the adaptive immune system. So that is the reason why adaptive immune system is going to do two things. Number one is going to fight against the enemy, eliminate the enemy. Then the second one is going to use it. In fact, majorly is going to use it as memory. It's going to store it in the brain, so that whenever somebody who is going to break their heart again comes, they will remember that, oh, they have come again. So that is the more reason. Naturally, we are being endowed to actually take care of whatever might be the immunological challenges that we see. And there is also a way we can also boost our immune system. Nutritionally, we can boost it. Apart from that, hereditarily, that is genetically speaking, there are some people that there are some certain diseases that cannot even affect them. They just discovered that part of Cameroon, there are some ethnic group that HIV, they just injected HIV into them, and HIV could not even do anything in them. Their system just eliminated it quite all right, and it's just within certain ethnic group. So it is very, very possible like that. So that's why nutritionally, genetically, and also environmentally, the environment in which you live will go a long way to determine your ability to be able to fight against certain infections. So if you are in the environment whereby the environment complement system are set of 27 proteins, that we call them cascade of proteins. And I told you the meaning of cascade. Cascade means that it's just like you line up some plastic. Let's say you line up different types of plastic that they touch each other. By the time